Right, my name is Sandeep. You can call me Sandy. Today I'll be talking to you on some of the concepts of determining indels generated by CRISPR-Cas9. This sort of fits in well in terms of how we can use these non-expensive tools to understand and play with your data and then use that information to also design your experiments. That's the whole point. I'll start sharing my screen now. We'll go through the presentation. We do have some practical demonstrations that I will show you on the screen, and I've got some data sets for you to play around with as well. So for the data sets, we've got three folders which are by the name SNP checks, knock-in checks, and tide analysis. So these three folders, if you can download and store it on your drive or on personal drive, it'll be useful to use some of the files from these folders during the testing sessions in your program, which is the SnapGene that you've been requested to download. As I mentioned before, I'm Sandy. I work as a technical specialist in the cellular operations team. So in Sanger, we sort of run huge pipelines to perform large-scale screening or even experiments to generate high-throughput data. So when we work in such projects, we always have to combine a sort of the most reasonable way to achieve high quality data in the most high throughput format. And that's where some of the things that I might talk about might come into play. And I've had a chance to work or with a few softwares, a few machines. I'm both a bench-based molecular biologist and also have some skills in bioinformatics. But as I say, I've tried to find the sweet spot between bench work and office work, sort of to understand what would be the best way to achieve these. You've already been talked a lot about gene editing by CRISPR, but we're more focusing on using the freeware tools that are available and to sort of understand how we can use the same tool to facilitate gene knock-ins or even tagging if you can. And what I also want this session to be an opportunity to understand that how CRISPR-Cas9 actually produces this indels and how it is important for you to understand what cells you're working with, what models you're working with, and we'll touch about it a bit later on that. I'm not going to go too much on to delve too long into the CRISPR-Cas9, but basically we know that CRISPR-Cas9 using the guide can generate double-strand break, and this generation of a double strand break has two consequences that the cell can, choose to repair itself. So one of them is by homology directed repair, which produces an exact strand of replica strand, correcting whatever has been damaged. The other one is using a non-homologous end joining repair, which can produce mismatches. Now, how we use these both is quite interesting, and in my sort of experience with the systems in the last 10 years, I have subtly manipulated one or more of these pathways to actually obtain what I really want at the end of mine. And that sort of gives me an advantage to use the same system that I would like to work with. Example, disease models, whether it is iPSCs, mouse epiblast stem cells, etc. But it allows me to use the same models that are readily available and for resources and just gently manipulate them to achieve what we want at the end. So in terms of non-homologous end joining, when the CRISPR-Cas9 system can be used to generate knockout lines, so what it creates there is a set of indels or insertions and deletions that are introduced as a result of this double strand break. And this is particularly useful for disrupting a gene or disrupting a part of the DNA sequence that you're intending to target. It's mainly beneficial for generating your knockout lines, and it's quite useful tool to actually disrupt even intra-exonic regions, which means you can alternate your genes to produce splice variants that have never been discovered or never been sort of understood upon clearly. So you can think about using this autonomous sort of result of insertions and deletions. And when we go through the slides and some of the data, you can sort of start to predict what indels can be generated and how you can use the system to generate your end product that you would want. In your other system where you would like to use your homology directed repair, it's quite useful in generating novel lines or reporter lines, and it's particularly useful for a knock-in system rather than a knockout system. What this means is you are using the CRISPR-Cas9 to generate a double-strand break, but forcing the cells to accept a donor template which has homologous recombination regions at either end. And the concept of that double-strand break being repaired provides an opportunity for this donor template to be integrated into the site of DNA. This way you can sort of perform site-specific insertions like fluorescent tags. You can modify promoters or regulatory sequences when you look into promoter regions of your exons. You can overexpress genes, you can repress them, and so on and so forth. Depending on which pathway that you would like this CRISPR-Cas9 to take, you can sort of achieve. There are pros and cons for both of them, and we won't be going into that, but there are a lot of synthetic chemicals that are available through various vendors, which can sort of stimulate the cells to use one of these pathways in compared to the other. That sort of gives you the opportunity to screen for less number of cells that intend to have your knock-in or knockout. The slide here actually tells you what sort of workflow that we use. We've gone through our guide RNA design and we have an optimized culture system. We tend to use a RNP complex, which is we don't tend to clone our guides in, and we just order them as single guide RNA, as previously discussed in one of the sessions, combine it with Cas9, and we introduce them into our cells. At this point, you've got the double strand break generating, and you're trying to actually screen the cells from an expected phenotype to the genotype that you actually want. Now, the simplest way to identify a successful genome editing is to observe the cell's phenotype itself. Some of the genes that you would target would be really crucial or essential, which means it causes a sort of differences in the cell culture pattern that you're working with. It sort of changes the cell's phenotype, and it's quite easily captured by just visualizing it under a microscope or sort of understanding if the plated cells that have undergone the gene editing actually survive that or not. This might not be that evident for many of the guide RNAs that you're working with, or many of the genes that you're working with. We therefore need to analyze them a bit more critically, and for that we need to go to the next step, which would be to understand what happens at the introduction of your guide RNA and Cas9 into the cells. So we look into the cell's DNA and sort of characterize the actual events that are happening in here, which tell us how efficient the gene editing has been. To do that, we've got a few methods which should be in this slide. So many of the techniques that some of you might already know this, but to determine the edits and subsequent effects in a cell type, we've got a few of them. Some of the methods that are here are just using NGS, so next generation sequencing. You've got tied or tracking of indels, which we will touch upon this afternoon. You've got the endonuclease mismatch assay, which is a lab-based assay or a T7 assay, and you've got the IDA method as well. What's common in these is that you are working around with your cell's DNA, working with the pool of cells from a genome editing experiment that you've done. And the first step, as you can see, that's common here is the performing of the PCR. So you would need some DNA to understand this, and you would definitely need to amplify it. What happens after that is a bit more process specific. You've got your NGS that can handle quite a lot of indel tracking in one run. Its sensitivity is quite high with a very high resolution, but sometimes the scalability is quite low because it's already high throughput. And one of the main things with that is a high cost. The other two assays that are really that are at the bottom, that endonuclease assay or the IDA, they do have quite a good, they have low resolution. It's not as high as the NGS, but their cost is quite low, thus making them one of the most easiest way to perform, sort of understand what if your phenotype has been affected with the guide. The middle one that we can touch upon will be a more of a medium-based assay, which is the tide. And what you can, what sort of happens in there is you'll be looking at a medium sensitivity and medium resolution of your cell pool. To those who have not done these mismatch assays or sort of understood or performed any gene editing, a survey nuclear assay or a mismatch assay is something that you can perform in the lab without the requirement of any software or any sort of bioinformatics tool. So it uses a nuclease assay, which is a family of mismatch endonucleases, mismatch-specific endonucleases. What it does is when you've got your genome edited pool from your targeted cells, and you have a modified locus in there, the first procedure you would do is to perform a PCR. Now, all those PCRs that I've been talking about in the previous slide amplify a range of DNA that do contain your locus or site of interest. What it then does is, because it's from an edited pieces of cell, edited pool of cell, you would denature and anneal your PCR fragments, giving it the opportunity of all the PCR fragments generated from these various pools to reanneal with each other. What I mean by that is, if you perform a PCR from a wild type cell, you will be just performing the natural amplification of the DNA while it's there. If you perform the PCR from the edited cell, you'll be amplifying that particular region. However, when you denature and anneal it, you give the conditions to actually create these mismatched fragments or double-stranded fragments, which then which then is recognized by these mismatch endonucleases, such as T7 endonuclease. Once you subject them to a restriction digestion using these endonucleases, what you generate is a PCR product that are fragmented when you incubate them with your endonuclease. So here, as you can see, you've got your untransfected cells, which are just non-edited cells. You've amplified your PCR fragment and you've performed this assay. It doesn't give you any sort of smaller fragments, indicating that there are no mismatches within that region where you amplified that product. However, when you look into sites that have been edited either by an example guide 1 or guide 2, it sort of generates these mismatches at a particular place in that PCR product, which the restriction enzyme or the endonuclease actually identifies and cuts it out. So this assay is entirely lab-based. All you would need to do is a PCR and run a gel to actually tell you how efficient your cutting or how efficient your editing has been in your pool of cells. It's been quite useful for us in terms of scaling down. The costs are quite sustainable. We can have the whole process from point A on the top to the point B is literally about 4 hours, 2 hours for PCR, half an hour for denaturing and running running the restriction enzymes. So you can do it in a day. You can be confident how well or how efficient your pool of cells have been edited. The second one, which also is cost is quite low and is quite a robust way of identifying indels, is indel amplification, indel detection by amplicon analysis, or IDA. It's proprietary based, where you've got a PCR based method, but it has an additional primer which is fluorescently tagged. So as you can see here, if you've got a sequence of DNA where you have a site-specific gene that needs to be knocked out, you would be performing a PCR with 2 primers, but then you will have a third primer which is fluorescently tagged that gives you fluorescently based amplicons that are generated at the end. And these amplicons from each of them have this fluorescent tag, and they are run through a detector. And this detector, as you can see, provides signals telling us which sort of, if you have a reference sequence, which would be from your untransfected cells, they will be sitting at a position with the highest or with no indels, and then you'll have allele sizes that appear either from a deletion of bases or from insertion of bases at that particular site. It's quite robust. I've had a chance to experience working with this protocol. It's really fast turnaround as well, and you can run up to 10,000 samples with IDA, and it provides you with confidence the indels that sort of are generated within 4 to 6 hours of your Cas9 generating those double strand breaks.
Ila has asked, can the EMC or T7 approach be considered quantitative?
Fantastic question. Absolutely, they can be quantitatively assessed. Let me go back to my slide so that I can explain a bit more. Thank you, Cassandra, for catching that. So in here, in this gel, if I zoom in a bit on my presentation, one thing you can do is you can quantify your PCR products before loading onto a gel. But for example purposes, say I've loaded equal amounts. If I have my untransfected, which that has not been cut by the enzyme and that has not been into the process, but I've also got the product that has gone through the enzyme digestion, you can see that there is a slightly thicker band here. In these two, where there are two guides, you can sort of use densitometry to understand how much proportion of this thick band is lost and how much of the other fragments are being generated. Comparison of this band, that is the correct wild type size, and loss of its fluorescence or intensity in comparison to the untransfected can give you a very good idea of how efficient your guide is. We've had some of our pools targeted by very efficient guides where you completely lose the top band and you end up with a series of fragments down, saying that that site has been completely demolished by that guide and Cas9, saying that that particular guide is very effective in generating a disruption to that site. What has happened in that site, it's still not it would not be clear. That is whether the bases have been inserted, whether the bases have been deleted, that you would not know. But what it can tell you quantitatively is that if if a if you completely lose that, it'll tell you that your guide is 180 to 100% efficient. If you have an image that is like this in this, it'll tell you that it's around 30 to 70% efficient, so on and so forth. The less intense your amplicons below this are, the less intense your guide is. I hope
I
that helps.
Yeah, I have a follow-up on this if that might be possible.
Yeah,
It's just that if the guide has off targets, you might not see them because here we amplify one specific region for the PCR. If the guide has targeted other places, and the wild type band is completely gone, and then you don't have enough on these two parts that have been cutted, it means like
I
it
I
has got
completely
some.
agree.
Yeah, it
I
has got
completely
somewhere
agree,
else.
but
Yeah.
but the point with that is you will be amplifying only your region where you're looking to
Yeah.
understand your
Yeah,
guide, no
that's the
matter
thing.
what, not whatever procedure you do. And I've done another course just a few days ago, and I'm going to be very frank with this. All guides do have some off targets.
Targets. Yeah.
All guides will have an activity that you will not know.
Mm.
Again, I should be in terms of being more sort of just open with this, is that it's the Cas9 that is actually making the double strand break. So the guide is just guiding the Cas9. So where it is binding is the site where your nuclease is going in and creating it.
Mm.
If you obviously look at one particular site, you will get one answer. If you look at a different site, you will get a different answer. And if you are looking for guide design, we've just recently I've looked at example guides recommended for a gene X by various companies that are available online, such as Synthago, IDT, etc. If you compare the top guides for each of them, there are they are not the same. That means each company have their own favorite guides, and all of them have different sort of off targets. So it's all a prediction based. So if you're looking to actually look into off targets, performing this with confidence would be a bit more difficult. It is very important when you do a pool
Yeah.
screen, but for an arrayed one, I think when you're looking into a very site specific thing to happen, we would be going in with the guide that works at that site. So I
Yeah.
will touch on that
Yeah.
in a bit
Yeah. Sure.
more
Sure.
as well.
Thank you.
So coming to the tracking of indels by decomposition. So there's a very straightforward method that uses PCR and Sanger sequencing, and there are freeware tools that are available. We'll touch upon that in a bit. But the reason I've given that we've sort of have this as a medium sensitivity is that it can miss and you are limited by your Sanger sequencing. That's the reason we can't look into longer sequences. Sanger sequencing usually takes around, I would say, 1.5 to 2 KB maximum of sequencing trace files that would come out. So you are looking at a very particular region, in fact, as suggested, and that sort of comes down to its resolution. And in terms of its resolution, sometimes you would not be picking out very sensitive changes that, for example, detecting a heterozygous would be entirely difficult from this pool. Whereas if you do that through NGS, the amount of reads that you get for that particular change is quite high and quite gives you that confidence to detect heterozygous clones in there. So it's a simple and accurate assay, and it's sort of designed to identify targeted mutations generated through any double-strand break. You can use this not just for CRISPR-Cas9. I've used it previously for other nucleases such as talons and zinc finger nucleases. You've got different programs that you can use to run this. So what it requires is a standard molecular biology reagents. What you need to do is perform a standard PCR reaction and send it for sequencing with one of the primers that you have. You would need a control sequence or a sequence from an untransfected cell line as well. This is one of the outputs that the TIDE results would show out. So you've got what you're seeing here are DNA trace files in here from a wild type sequence, and from a mixed, if you do the same from a mixed pool of edited cells, after a site where your guide is supposed to be present, you will see all these mismatches coming in. What TIDE does is it deconvolutes each base, each read from that pool of cells, giving you all these mixed reads that are available for us to interpret. And it sort of plots it in a way that it tells you how many of these reads have an insertion at the first base, how many of these have a deletion at the first base or at the fourth base, and so on and so forth. We'll have a look at this in a minute, and it also tells you what sort of break site for that particular guide is given, and what efficiency that is your experiment, is the guide working in your particular experiment. So for your TIDE analysis, you've got your folders, which I'll show you now. I'm going to stop sharing my screen at the moment and change it to the web browser.
One quick question.
So yeah, Kani has
Yeah, sorry, I was just going to ask it out loud, but
yeah.
does it TIDE work for substitutions, or is it only insertions and deletions?
I will show you it online with one of these tools. It's easy to interpret there. So, okay. So you've got your TIDE. In here, if you see here, we've got your TIDE, which is just the decompositions of the indels. The program on the left has been generated like many years ago, and that tells you about the insertions and deletions. It tells you about the substitutions as well, but you will have to deconvolute that idea in that sort of result in your trace files. On the right-hand side, you've got another TIDE variant, which is TIDE recombination, and that's quite important for generating homologous recombination events in your experiment. And we will touch upon this in a few later slides. So if you go to Start TIDE, you would need three things, which would be your guide sequence, your control trace file, or the file that you obtain from your Sanger sequencing, and your test trace file. In the folder that I shared before, or which is on the LMS, you will have if you go into TIDE analysis, you've got about 8 sets of data in there. And in each of the set, you've got 2 trace files, a control and a sample, and a guide sequence. You can copy that guide sequence as it is and load it in here. It's just a paste. And let me say that this is, and then either you can simply drag your trace files into its correct location. So I've just dragged my control sample to the control tab and my test sample to the sample tab. Usually, as soon as you do this, the program automatically gives you what has happened or what sort of data you're looking at. Now, you need the guide sequence to be 20 nucleotides only. You don't need to include the PAM. If I include the PAM, for example, CCCGG in here, it'll automatically tell you that it should only be 20 nucleotides. On the first graph that it sort of is giving you is a editing score for your test sample that is here in comparison to your control. What it tells you is the effectiveness of how well your guide has worked. So you've got a total effective score of about 46% of editing. If you look into the graph that is here, you can see that it starts with 0 in the middle, and you have insertions on the right and deletions on the left. The way you read this graph is that 41% of the pool of cells in your test sample still are the wild type sequence. That means they are not edited. 15% of your pool of cells from your test sample have a single base addition. So we still don't know what base it is. It can be A, T, G, or C. And what we're looking to find is that 15% of that population has a single base pair addition at that particular place. Similarly, if you look into 12% of the remaining population, you have 2 bases that are being deleted, and you've got 6 bases that are being deleted in the 14% of population. That's the overall editing score that you would sort of interpret from such a result. So the red bars are tell you that it's a very significant, and it's a statistically probable event that all the cells have these sort of, or the proportion of cells have these sort of deletions and insertions. There are a few that have been detected which have a slightly lower score. You can scroll down and you can update your view, or you can look into your advanced setting and tell it to see a bit more further. So if you can extend the boundaries a bit more, if you have a very sort of effective sample, it'll give you what sort of windows you're looking at. If I look into the indel range, I'm only looking at 10 basis deletions on the left and 10 basis deletions on the right. If I say that I'm trying to look for bigger deletions and bigger insertions, it'll still try to find a bit more what is happening far away from the guide site that you're looking into. This is quite important because many of the work that we do, or sort of designed for gene knockouts, is that you will have multiple guides that will sit on an exon a few bases upstream or downstream of each other. And it is advisable that you have your decomposition or indel size range window to be maximum to see the effects or co-effects of these two guides. So particularly this particular guide is sort of having an effect where the double strand break is recombining the events and sort of inserting one of the bases at 45 after 45 bases in the genome. How it's doing that, it still doesn't know. But the more you look, and as you can see here, you've changed your efficacy from 46 to 47%. The bigger you look, the more information you will find using these. So I'm going to just add in another example now from one of the sets that I have. So I might be looking at set number 7. So I've got my guide sequence for set number 7, and I'm just adding my trace files. So once you do that, you would need to reset your boundaries so that it can start performing its range a bit more stringently than before. So if I look into your decomposition window, now this particular guide and this particular samples tells me that it's quite inefficient. It has almost, that particular pool of cells has only 70% that have remained wild type, and you've got only 27% that have been effectively edited. As you can see here, this sample has more deletions, or more sort of deletions that have been possible, but less of insertions. And what I want you to have a look here is that if I increase the indel size range, sometimes it doesn't really change that much. For an ineffective guide, if your experiment is not that sort of robust, or if your guide is not that effective in conducting gene edits, the more further you look, you won't find that anyway, because you will be working with the wild type sequencing on its own. Before I sort of give you a chance to perform this, I will be going into using SnapGene to actually tell you how you can use the trace files to do this analysis on yourself. So going back to my first example, where I've got my guide sequence here. So just copying it. And I'm just adding my trace files as well again. Okay, so you've got your results back again. I'm going to stop sharing my screen and just work on showing you an example on my desktop actually. So for Kayani, you wanted to know what substitutions or how you can determine. So you've got programs as Shalini mentioned that can show this, but that is set 1 that I've looked into. And if I open these trace files using SnapGene, what you would find is a sequence read, as I've shown you in my slide. So you've got a very good read of DNA sequence from your PCR product. It will be ending at one point because that's how much PCR product we have. So it doesn't read after a point. That's my wild type sequence. I'm also opening my trace file for my test sequence and aligning it down there. So what I can do is, if I have my guide sequence, the region which I want to assess, I can simply copy it in my sort of, control C format. I can go to my control trace file and find it by pasting it there. If I paste it there, it tells me that it's around the region of 280 to 300 bases of the sequencing file that my guide site is. And if I do the same in my test file, you can see that I have detected that guide. And in here, if I scroll on the right, I can actually see what substitutions it's doing. So I have a C here, which is entirely substituted evenly with the other bases. A more prominent one, you can therefore start to walk one by one or base by base. I can see the T that is here is entirely substituted by other bases, and so on and so forth. And that is what your tide is actually telling you. It also tells you that there are 5 bases that are deleted and around 6 bases that are deleted. So we can have a look if we can detect that as well. So this is to the left of the guide site. So we need to, I think it's looking around these areas just around the guide site. So in this way, you can start to look into what sort of deletions and substitutions it has done. And that provides us with an opportunity. If you want to actually get an idea of how each of them, or what sort of things it has sort of manipulated within that particular experiment, doing an NGS will give you a far more better resolution than your tide analysis. The tide analysis, or even this particular analysis, just finding a site and telling you whether it's edited or not, it gives you confidence that the pool of cells you're working with has undergone genome editing and you're proceeding to look into the next step. As a short exercise, what I would like you to do is, I've given you 6 sets or even 8 sets of data in that folder. If you guys want to have a go for about 2-3 minutes, you can have a look. I would recommend you to try this with at least 2 sets from between sets 1 and 6, because if you see the guide sequence, it's exactly the same sequence that I've used. What sets 1 to 6 contain is a particular experiment that I've done across various cell lines. So these contain hex cell lines, iPSCs, colorectal cancer cell lines, and you can see how the guide actually produces different indels in different cell lines. So you can't expect the guide to work exactly the same in each cell line. So that's you're welcome to have a look at it for about, I would give around 4 to 5 minutes for you guys, and I'm happy to take any questions. And if you feel stuck, please let me know and I will run it through the tide online. I'll keep the Zoom share on.
May I ask you one thing? Do we have to
Go
open a
ahead.
snap screen and tie them together?
No, you can try either of them. You don't need the SnapGene. The SnapGene is to tell you that you can still look at the trace files and make a coherent, informative guess of how the tide is working. But I think I would recommend you to trial inputting your samples in this and see how different it is, especially with the different cell lines. It was quite interesting to find that the guide, even though it's sort of taking your Cas9 nuclease to the exact location, what the Cas9 nuclease is doing is entirely different. So I think that was quite informative for us as well.
Sorry, just remind me, we don't play around with the decomposition window values, we just keep it at maximum, right?
Yeah, you can. You are welcome to play around. It will give you different sort of it will give you different answers. But try it. It's more of the more you work with it, the more you will understand. So sometimes you need the decomposition value to be around certain length, depending on how much you've sequenced, and that sort of works around for different samples. So try ahead and see what you get down for.
Sandeep, I have a question.
Yeah. Yeah, go ahead.
So, okay, I'm just playing around with the download files, but to understand if indeed the guide RNA that we used is a good one? Which editing efficiency we should expect?
So I think I would say anything that is above 50 to 60% is quite good. It does depend on your cell line. It depends on your delivery as well. So in terms of delivery of Cas9, I think, and in certain terms of cell line, in cancer cell lines, I would expect the guides to be efficient above 70%. That's something that can generate a double strand break, and you can use it for knock-ins, knockouts, whatever. For difficult cell lines like iPSCs or embryonic stem cells, etc., I think a good guide would still be at least 40 to 60% efficient. So,
Dr. Sandeep,
yeah,
I was checking around the set 3,
yeah,
and set 3 has 2 forward files, sequencing files.
yeah. So I think one of them is an SCF file, which is still be acceptable. So I'm loading set 3 as well into my
I tried
screen
both,
here.
and both show different results.
Yes, that's because, let me just check it again.
Yeah, you can try.
So the SCF file is not uploading on mine, but I'll try the AP one if that's working.
Okay.
Your guide sequence has been deleted. You need to put guide sequence back in.
Oh, thanks. Good find.
Regarding the efficiency, Sandy, can I add one point?
Yeah.
What I noticed is that when we do ICE analysis, and if it gives 100% efficiency, and we compare it with the NGS, it'll be around 90%. So NGS is more gives you more accurate. But I haven't used TIDE, but yeah, I think it's TIDE and ICE are similar. So, but they give you like more efficient. It gives you somewhat like 10% more than the NGS.
Yeah. Okay. Thanks. So you've asked the question. So again, in my web browser, it's taking a long time for the tide to work around. Perhaps everyone is working, but I've skipped tide and I've just started to look at the sequence as it is for particularly set 3. So for set 3, what I've seen is if I try to find the guide sequence in the set 3, it doesn't really find. And that sort of is a problem because it thinks that the site is being lost in your samples. That's because your PAM sequence is actually edited in this cell line. So, or not the PAM, just the last 3 bases. If I remove 3 bases, it still detects exactly where your guide is supposed to be. And you can see on the right, it's generated a lot of indels. So it's a very efficient guide, particularly in this cell line, and that sort of has generated a bit of convoluted data for tide sometimes. It does happen. It's not entirely foolproof. If I look into the other file, which is the SCF file, it might be the same issue as well. So I can't find my guide completely, but I tried to trim it down from one of the ends, and I think it's still probably not able to find it. I think it's a sort of a different run that I've done. So that's the reason you get different sort of indels.
Interestingly,
Okay.
set three for me
Yeah.
worked, but I had to use the AB1 files on both.
Yeah, yeah. We're going to continue with our analysis later on. So one thing I wanted to say is that jumping back into the presentation, we've got your wild type and your mixed pool, right? Now, what needs to be done is that when you send your samples from wild type and your mixed pool of edited cells, you need the same primer for the sequencing to run. TIDE cannot understand if you run the wild type on a forward strand or a forward primer and then try to identify the indels on the reverse. It will tell you that I can't find that sequence. Okay, so if you look into the TIDE website, there are a few set of recommended rules. So you need the guide RNA, the wild type, and your test pool to be in the same orientation. You can use all 3 in the reverse orientation, and it'll still give you the same result, because what it's looking is a double strand break rather than what strand it is. It's just a general strand break. So because it's a double strand break, it should generate the same sort of results whether you look at it on the forward or on the reverse strand. And it's a PCR, so all strands are present there, being a double-stranded amplification. Touching in on a few other, so we've looked into what happens with the non-homologous end joining, how simple it is actually to understand what cell lines you're working with, what guide, or how efficient it can be. These sequencing runs cost around £2 to £3 per reaction. So if you have a few genes worth, all this data can be estimated by within like £10–15 worth of sequencing runs. So it's not that costly. It's very cost effective. You can use these similar freeware tools to actually understand your knock-in as well. So in generating a knock-in, and this is why I was saying that when you're working in a very particular array format, I think identifying the best guide, identifying the off targets, those sort of things become a bit more secondary. What I mean by that is we've in our end we sort of worked on generation of homology directed repair or HDR libraries, where you will be learning this later in one of the sessions anyway, where we are looking for Cas9 induced double strand break to generate base pair changes at a particular site. Now, if, for example, in here, I'm using CRISPR Cas9 to generate a double strand break, and then I'm providing a donor template, but I'm only changing a single nucleotide at a particular location. So if my nucleotide is a G at that particular point, I would like to substitute this with a C, with a A, and with a T. That is completely achievable by providing 3 different HDR templates during the experiment, which then allows me to recombine at least one of them at that particular site. For such a process to occur, I cannot find a guide that would be cutting elsewhere. I need a double strand break, particularly at that junction. If I need to create a knock-in at a particular site or perform a double strand break at a particular region, I cannot look for the best guides with the least off targets, so on and so forth. I would be using whatever is available around that sequence as my benchmark to actually perform this, and I would be relying on a very less chance for this to happen, but the probability is it will happen, and that's what you're basing it on. So in a gene knock-in experiment, in generation of cell lines, in generation of fluorescent reporter tags, in generation of single nucleotide variants, it's all about the site where you're generating that double strand break, and you would not be relying on guides that are highly effective. You would still work, if possible, work with the potential of pair of guides simultaneously, or even 3 guides simultaneously, but you would try to create that pressure on the genome by inducing the break at that particular location and then providing with a template to recombine and insert your thing. So here in this particular example, we are having homologous DNA alongside each of these, and we are changing one particular base at that particular location. Now, if I do this for the way I do this if I want to create a knock-in is that I would have 2 guides, one with the guide RNA on the reverse strand and one with the guide RNA on a forward strand, and I can create a knock-in using a fluorescent reporter gene such as GFP, etc., by opening up the genome at that particular locus and allowing the homology to insert your fluorescent tag in there. So this has been quite useful for generation of different cell lines, and the way you would do this is that you would be looking into the homology directed repair and pushing cells to use this mechanism as compared to the more efficient NHEJ repair mechanism. Now, pushing cells this comes at a cost because cells do not want to do this. They want to go to the easy part where they repair this double strand break quite readily by just inserting and deleting some of the bases. So therefore, finding your insert is a bit more harder sometimes, and you would need to do a thorough QC. So one of the examples that we'll look at now would be your SNP change. So you've got your genomic DNA where you want to perform a site of alteration. What you need to do is, as a donor template, you would be looking to have some sequence of the wild type DNA flanking your site of alteration, and these are called the homology arms. So they are quite important for this process. And where it comes is because it uses your donor arm template as one of the arms to repair your DNA. And because it matches your site of alteration, it uses this as a template, thereby inevitably adding in your sort of genomic DNA of interest at that particular site. So you're you're sort of tricking your cell's repair mechanism to use your donor template as a potential repair template to repair that double strand break, thereby tricking it to insert it in that process at that particular region. That's the way you would be looking to infer this. Because it's a bit hard, you would need to perform a lot of primary and preliminary QCs at this, and it is always important that you work with single clones when you perform these sort of edits into your cells. When we looked at the tide analysis in the previous slides, we were working on a pool of cells. So we would then go on to scrutinize that pool of cells and start to work with single sort of cells when you come to generate these models. The indel tacking is quite easy. Now, I did mention that Tide has another sister sort of program, which tells you how much efficiently your cells can recombine. And you can do this by the ICE analysis, as Shalini mentioned a few minutes ago. ICE analysis is a program given by Synthego or EditCo now, and it tells you, it sort of follows the same concept of putting in the guide, putting in a donor template, etc. What I'm going to show you as an example is in here is a DNA sequence. It's a snapshot from the program SnapGene, where we want to create a mutation at that particular site. And for that, I've used 2 guide RNAs that are very local to that site. The further away I go, the chances of me doing that particular or obtaining that particular mutation will be a bit more difficult. As you can see here, for this particular mutation to occur, I've generated this single oligo donor that is highlighted in the purple color. You can see that the particular mutation highlighted here is almost at the center of that donor fragment, giving me an opportunity to use this as a homology arm on either side. We will look into what sort of recombination events that this can happen, or this sort of design can provide us in the next slide, and we will be using EditCo for that. So I'm just going to go back online. So for this particular exercise, again, the folder in LMS you would use is not your tile analysis, but your SNP checks. Okay. So in terms of your design and guides, I would be looking into the guides and primers file, which is this one. Let me just remove this. So that's my design element that I have. So the first thing that you will need to add here is, let's see, from my design element. So it's exon 13. So I'll try exon 13 test. I've got my Cas9. So it has a lot of nucleases that you can use, but we'll use Cas9 for this. It tells you that you can add in both guide RNAs simultaneously for that particular program. So all I do is go to my sequence. I would take my guide RNA1, copy it. Oh, it does tell you that do not add PAM sequence. So I'm going to remove the PAM from here. So just 20 bases. You can also add the other one. So if you see my design here, it's on the reverse strand. So if you're using SnapGene, what you can do is you can click this, or you can just use 20 bases just before the PAM sequence. And what you need to do is you can copy the bottom strands from this, which is the edit button here. So, okay, copying my bottom strand. The donor sequence is optional, but I would be using this because what we are doing is we are not trying to find our insertions and deletions. We are trying to find recombination events. So the donor sequence is the purple donor sequence that I have here. I would just copy it as it is, and I can paste the sequence directly into there. And from the trace files, I would then go to your exon 13 wild type, which is the control AB1 file. And I would go to your exon 13 test, which is the test file. I would add this to my analysis, which means it only adds it down below. So I do have another gene, which is, or another site, which is the exon 21. I'm going to do the same. And this way you can have a better throughput in just one single run. So I'm going to do the same, just copy my guides in, add in here. I'm going to write this as exon 21 test. I'm going to take my donor, add it to my file here. I'm just using one of them. You can add the other two as well if you would like to. Just I'm going to drop my wild type sequence and my test sequence here as well. So this particular program allows you to run up to 96 or even 200 samples at the same time. And once you've loaded all of them, you can even upload them using Excel file or drag and drop, etc., etc. And it's quite a useful tool, quite a useful freeware tool, I would say. And you can do this without the need of any bioinformatics knowledge. It gives you quite a bit of understanding. So it's analyzed for the first sample in here, which is exon 21. When I click on it, it tells you what does it mean. It says that it's a good edit for that particular exon. It tells you what the PAM sequence is. It also tells you if it's generating any indels or any knock-in scores in here. If you don't have any donor and alignment, it'll tell you that if your recombination has been poor, or if your recombination has not been that successful, and so on and so forth. So you have a lot of edits that are happening, which is generated by here, but it's not yet aligned with your knock-ins at this point, and it's quite a useful tool, and you would be getting this the same when you do the tide as well. If I go back to my runs, let me see if I can find it. And what I then do is, instead of putting in just one guide sequence, if I put two, exactly how I've done the experiment, sort of a thing. So in the first instance, we used just the first guide that was away from the site of mutation that we wanted to put in. This time we added both guides in to see if it actually gives you any difference in terms of understanding what it's interpreting from that particular run. So just wait for a couple.
The annotations you have in your files, they're annotations that you've added up. That's not how it's come off the sequencer, right? Sorry, I've never used Snapgene.
Yes, that's
before,
not. Yeah, you're right. That's just the annotations that's added physically, or on there. It's not how it's sequenced.
And they won't interfere. They won't interfere with edit code if you upload that version.
No, they don't. No, they should not. They will not.
Which is the donor sequence that we have to insert?
Oh, the donor sequence. Sorry, the donor sequence is the purple sequence, which is single strand oligonucleotide donor that is short in here. That's what you would need to insert.
So just to clarify, that is a Snapgene file, correct?
Yeah, those are Snapgene files. Yeah. So one of the models will show you if there are any nucleases or how the indel distribution has been. I will have a look again in a few moments to see if you've got any sort of recombination events that are getting there. So it marks you the two guides that you're trying to validate and sort of edit it if there are any sort of knock-ins that you can get from your edited samples. That sort of tells you the recombination events as well. Let me go back to my presentation and I'll see if I can show you how that sort of thing works. So if you get a knock-in score, so this I've done for the same guides again. So these are the two guides that we use for exon 21, and you can see that it's generated an indel score of 36 with a knockout score of 27. It means that it's created a lot of knockouts in there, but whether it's recombined or not, it'll tell you by analyzing the recombination events, and that's what I wanted to prove it at. At these data, I think you can have a look at how this would sort of make you understand. Sometimes it's quite difficult to obtain any generation of a knock-in at a particular site, and it's quite important because we are here just performing a single nucleotide change with a single stranded oligonucleotide donor. Coming to the other part, where you will be using fluorescent tags or generating a fluorescent tag for a reported gene, that is quite efficiently managed using CRISPR-Cas9, because you've got your same site of alterations with two homology arms, and what you're now looking is to insert a particular fluorescent tag at a particular locus using CRISPR-Cas9. And when you create these cell lines with your reporter tags, you can actually use flow cytometry to tell you how efficiently that these tags are generated, and whether your single clones that you pick from these pool of edited cells are actually fluorescing or not. Going back, I would like to use SnapGene to actually show you how this would work. And the last folder that we will use is our knock-in checks, where you've got your genomic DLX5 DNA that is here. So you've got your genomic DNA here, and because I wanted to knock in a fluorescent protein at that particular site, again, I would be not following the rules of efficient guide identification. So that's the translated region of a gene that I've marked with, highlighted with, and I want to insert my tag right at that end. And I would be using the guide sequences that are either overlapping that particular site or very close to that region where I want to conduct that recombination. And for that, what we can do is, using SnapGene, you can see the model that I've sort of created, and these are again available for you to have a look at in your leisure, where you've used that particular site to open up your gene and insert this M scarlet in here. When you use your Sanger sequencing, it is advisable to sort of overlap these regions and sequence these regions using various primers. And this is what I wanted to show using your trace files that are available in your folder. So if you're opening this on SnapGene, what you would need to do is, if you go to the last tab down here, you can use the Show Alignments tab, and it opens up a portal where you can add in or import your sequences. When you import your sequences, you need to import the trace files that are available in your folder in there. And what they tell you is that they provide you with sequence traces that overlap that particular region, confirming that the sequences are indeed overlapping along with your knocked-in gene, which is your M scarlet. It's a form of a fluorescent protein as well. So these blue arrows tell you that it has been successfully integrated, and sort of theoretically you have tagged your gene with your M scarlet fluorescent protein. It will detect some of the changes that occur in your samples, for example, a single base pair change, etc., which you can screen when you're working with different sort of clones. Now, if I do the same for my wild type sequence, which is the previous genomic DNA that I showed you with, so this is just the exon 3 where the guide sites are. If I align the same trace files with them, they would align a bit, but what it does, it actually creates a bit of mismatch because it contains an M scarlet trace inside it, and that you can capture it using your SnapGene file as well. So all the sequence that you're seeing on screen, where there is no overlap with your context sequence, it tells you that it's a tag that has been inserted, and that tag is actually M scarlet. Obviously, when you're screening for clones and colonies, you can just screen these cells with the fluorescent tag using flow cytometry. And in here, in one of the PDFs that I've attached there, you can see that the wild type cells, when screened for fluorescent tag, do not show any positive cells for M scarlet in this particular plot. But the same tagged line, if I use it for your M scarlet channel, which is the red, you can see that all the cells that are there have indeed been tagged for that particular gene. So you can use CRISPR-Cas9 to actually generate these cell models quite efficiently and perform these recombination events to suit your needs. And that was the main sort of part of it, whether you want to create knockouts or whether you want to create knock-ins, you can use the system and an array of online tools that are readily available. You can skip the QCs in the lab and depend solely on sequencing of these lines, which generally uses a PCR product and a pair of primers, and see how well your experiment has worked. And hopefully we've sort of also briefly discussed the data coming out for Sanger sequencing. If you've got to time around with it, please do feel free to have a look into the files there, and you can actually open them without the need of these freewares. You can open them using Snapgene. Sometimes it might be even you can open it on Word as well. That's what I used to do before. And you can run through the sequence manually to see if you've got your correct gene tagged in. It sort of gives you confidence. The major important thing is that the guide RNA is just a tool for the Cas9 to go locate and start breaking the DNA. The different cell lines that you use will give you different results. The different phenotype of cells, for example, pluripotent stem cells, differentiated neurons, cancerous cells, all these will give you different results at the end of the day. So you can choose to work with multiple guides that you have, and it will provide a sort of an array of results that you would do. Cassandra, what
So
look?
there's just a comment in the chat, mentioning that, well, basically asking if the alignment option is available in the free version of Snapgene.
It used to be available. I don't know if they've changed it. I have apologies if that is not the case, but
It's not
Yeah,
included.
yeah.
you can use alignment tools. There is another program called DNA Star that will process all these files. So any file that come out of sequencing will come out as an SCF file or an AP1 file, and you can use other programs that can open up these files for you and use it the same way. The Snapgene is just one of the best tools for molecular biology work, and I've used it quite regularly, and that's why I've put it. But I do agree they have sort of reduced the number of features that the free version can do over the years, and it's a bit of a surprise to me as well, because Sanger sequencing and QCing was a part of a routine thing that we would do using Snapgene, but it's a surprise that even we can't do it with the freeware at the moment. Yes, of course, Benchling is also a great tool to use for these purposes.
Hi, Sandeep. Yeah, I downloaded the files, but all the files, set one, it's guide sequence, and control and sample is not even text, it's like picture.
So what program is it opening with then? So the extension file.
Snapgene, it's opening Snapgene.
Yeah. So it should be an application file. So the files that you would download is .ab1, and it should open in Snapgene. But even if you download it, try dragging them into the tide and see if it actually opens, as long as the application or the extension file is AB1.
Thanks.